really excited about the year of remote sensing. So yeah, I'm going to start with this picture of a boreal forest with a rainbow because for a couple of reasons. One of the things it reminds me of how the interaction of light and matter can inform us. In this case, the forest can inform us about the function of the forest. And the other thing is this looks like a fairly uniform forest, but there's a fair amount of functional diversity present in any landscape like this. And so that's something I want to address with remote sensing applications in the context of the flux tower footprint. Next slide, please. So my talk today will be about remote sensing, but remote sensing across a range of temporal and spatial scales, from very fine scale proximal remote sensing operating on short time spans, small scale, up to aircraft and then satellites at very large scales, global scales, and long temporal scales. So the processes, the biological processes, photosynthesis or GPP or carbon fluxes, carbon cycle questions that we're looking at are, the terminology varies, the details sometimes vary, but to some extent with remote sensing, these processes, the information can scale, which is very helpful. We can upscale or downscale. But one thing to realize is you have to be conscious of the scale that you're operating, because each scale has emergent properties that informs the next scale. So details can be a little tricky. So it's important to be conscious of the spatial and temporal scale that we're operating under. So that's a theme I want to emphasize right up front. Next slide. So I also want to give a shout out to SpecNet, and I think some of the SpecNet board members might be attending today. Fred Humrick is the leader of SpecNet at the moment. Lauren Albert, Danette Thayer, Ran Wang. And I want to encourage you, if you're interested in these topics, to stay involved or reach out to SpecNet. We have periodic meetings on the topic of integrating remote sensing with fluxes. And the particular issue is with flux towers, we have a certain footprint, and that's shown as the blue blob on the bottom. That's supposed to represent the flux tower footprint, sampling through time a particular spot on the Earth. And then with remote sensing, let's say satellite remote sensing, you have snapshots that might be covered as a single pixel, as shown in green, maybe a spatial snapshot. We may have time series, but sometimes we don't have really good time series. So how do you match the temporal time series of the flux tower with the spatial pattern of remote sensing? That's always been a challenge, and integrating the temporal and spatial domains across these different technologies. So the solution that SpecNet proposed almost 20 years ago is to put optical sensors in the footprint of the flux tower. And a lot of people are doing this now, and I think it's advancing our knowledge of how to integrate remote sensing with fluxes. And if you want more information about SpecNet, the link is at the bottom, and I encourage you to get involved or look at what we're doing if you're interested. Next slide, please. So how do we do proximal remote sensing? Well, it's an alphabet soup of different methods. You can walk around and sample. You can put things on towers. You can put things on mobile trams or on UAVs. And all of this is under the atmosphere proximal remote sensing. There are all kinds of other ways, including some I'll talk about a little later in this talk. But this is both one of the opportunities and challenges. It's an opportunity because we learn a lot from doing these different methods, but the challenge is this is non-standard methodology. So unlike the flux community, it has managed to come up with pretty good standardization of equipment and procedures. That hasn't really happened in the remote sensing world. So that's a remaining challenge. Next slide, please. So some of the questions I want to address, you can hit the first, how do we best integrate flux and optical sampling in space and time? And the answer I would propose is that, you need to have a mechanistic understanding of operational scales of key processes. So statistical methods are useful, but it's good to have a functional or mechanistic understanding of the basic processes. Is there a best way to do remote sensing? You often hear, oh, this method is better than that method. And I would argue that there isn't a best way, that these different methods are complementary, and you have to pay attention to the temporal and spatial scale of your measurements to make sense out of this and to really understand what you're doing. And that's sometimes lacking. And can we integrate our understanding of photosynthesis and productivity with functional diversity? And I would argue that productivity and functional diversity are intimately related, and it's important to look at them together. I think this is a way we could advance, the flux community, integrating remote sensing to look at the diversity within the footprint, for example, over time. And then finally, what future directions might be beneficial. And there's lots of ideas, but one of the points that I want to leave you with is if we integrate our methods across scales and integrate, for example, solar-induced fluorescence with the light use efficiency model across scales, I think that's going to be a huge advance. So next slide, please. These are my take-home messages. I'm kind of giving some of them up front. So I'm going to start off with a bit of a brief history of remote sensing, just to remind us that we've been doing this stuff for about 50 years. It's a long history. The idea of a greenness index was first proposed by Rouse in 1973, and the idea that you can use the red and the near infrared band to get a vegetation index that tells you about green biomass, leaf area index, or absorbed radiation by green material goes way, way back. And the NDVI is a good example. Next. And as far back as 1985, Goward and Dye showed that if you take time-integrated NDVI on a yearly time step and compare that to biome productivity across North American biomes, you get an excellent relationship. And this was a long, long time ago. So why is it that this works so well? A lot of people think NDVI doesn't work very well for looking at productivity. Well, it depends on how you use it. And it is, under the right circumstances, extremely useful. And I would argue that this is about as good as some of the best methods we have today when you look at large time scales and large spatial scales. Next slide. So along comes MODIS in around 2000, and this was amazing because it was designed with biospheric sampling in mind, covering the whole Earth with aqua and terra sensors. But it was also amazing because NASA designed a data system that would make the data fully available, including GPP products, NDVI products. And this was a huge accomplishment, which has led to a lot of amazing research. A little bit I'll mention right now. Next slide, please. One example from Steve Running's work, who produced the Mod 17 product, the GPP product, NPP, is this picture of NPP anomalies in the 2000 to 2009 period. This is actually Zhao and Running. So in their paper, they showed that there are areas that were declining in productivity and areas that were increasing productivity. So we start to get this picture of the dynamics of photosynthesis or carbon fluxes across the planet, and can attribute these, in this case, the red areas, to drought in this paper. Next slide. And, this is an interesting part of the paper, is where they compared the NPP anomaly, the red or the green areas, to the inverted atmospheric CO2. And it's inverted just to show the alignment of these two data sets. And you can see that the real effect of the carbon cycle, the fast carbon cycle, the biosphere, on the atmosphere in these year-to-year anomalies that are so characteristic of our atmospheric CO2. So we're starting to really understand the Earth as a system by putting this all together. Next slide. So one of the limitations of Mod 17 and the MODIS products, the MODIS GPP or NPP product, is that when you compare it to actual flux tower sites, it often doesn't compare all that well. If you look at individual sites, the relationship sometimes is not very good. Sometimes it is, but sometimes it's pretty poor. And it's not as good as we would like. So across different biomes, the global products do not always match the site-based products. So why is that? Next slide. Well, I'm going to remind you that this model is based on GPP, which is based, according to the light use efficiency model, on absorbed light and the efficiency with which that absorbed light is converted to fixed carbon. Next slide, or next Okay, and NDVI is key to this because it's really measuring the absorbed light. Next, in the Mod 17 product, the temperature and moisture effect on efficiency was defined through or parameterized through meteorological data, interpolated meteorological data, which is not at the same spatial and temporal scale necessarily as the satellite measurements. So you have a huge problem trying to combine two different data sets, which I would argue is not the best way to do this. And this is one of the main reasons why there's some issues with these products. People have been critical of these products, but I think they're actually pretty good. The model's pretty good. The parameterization could be better. And so the idea is, what if we could get all this information directly from remote sensing? Next slide. Why does NDVI work? Well, the understanding of this is pretty fundamental and goes back a long ways. This is something that Chris Field talked about in 1991 in a book chapter that he wrote. And the references are on the bottom, by the way. So, photosynthesis drives green biomass production, what we call the fraction of absorbed radiation by green material, or FAPAR green. Some people call that FAPAR chlorophyll. It has different terms. And that in turn absorbs the radiation that then drives the photosynthesis. So there's a feedback to the canopy or stand photosynthesis by this term in the middle. And NDVI measures this term in the middle, which is both the cause and the product of these processes. So it's sort of a circular argument. And this is one of the reasons why NDVI, when you integrate it across time and space, tends to be a pretty good measure of integrated biomass production. It's just when you look at the shorter timescales that can fall apart. Next slide. So this is just an example of a relationship between NDVI and FAPAR for a couple different vegetation types. There's many relationships like this, and depending on the exact data set and what range you want to fit your data to, it's either a nonlinear or almost a linear fit. It's a pretty good relationship when you measure it the right way. And a lot of people don't measure this directly, and I think that's another problem with the Mod 17 product, is it's derived through models rather than measurements. Next slide. So what can we conclude from this? Well, we have the tools for an integrated understanding of the Earth as a system. We see broad patterns and trends, changing seasonal cycles, contrasting biome responses, effects of stress, changing ecosystem atmosphere feedbacks like the drought example that I showed you. And but sometimes we get really poor agreement with individual flux tower sites, and the mechanistic details of these problems kind of are buried at this scale. So we really need to dig in a little bit more to understand this. And I would argue the model is good, but the parameterization is the problem. Next. So we see this problem particularly with evergreen ecosystems. I'm going to use evergreens as an example of a type of vegetation that when you look at the GPP, it goes up and down with the season. This is a boreal forest, or this example in the particular graph is from Pacific Northwest, but so I'm mixing my forests here. But what we see with NDVI is a pretty much complete failure to track the GPP. That's because the evergreens are always green, but temperature and environmental conditions change, and there's downregulation going on in wintertime that is not visible to NDVI. So what do we do about this? Next slide. Is there a better way to assess the efficiency term? We've got the NDVI, the plants are responding to temperature and moisture. Our parameterization might be not the best because we're mixing data sources. What if we could derive efficiency directly from the MODIS data? Well, it turns out you kind of can. Next slide. And here I want to point out the NDVI bands in red are bands 1 and 2. These are the terrestrial bands that have been provided on land for 20 years. There's a separate set of ocean bands that have been provided to the ocean community in blue, one of them band 11, that is particularly interesting to me anyway, and it's used in the chlorophyll carotenoid index, which has also been called the MODIS PRI because it's the bands 1 and band 11 together is the closest combination probably to the PRI index that I'll be talking about as well. But so this is a carotenoid chlorophyll pool size or ratio index. And when you add ocean bands to the terrestrial bands, we add a lot of information that is useful to assessing efficiency and also different functional properties of vegetation. Now, this isn't hyperspectral, but it's moving us a lot closer towards that because we have enough information here to fill in some of the missing parts of that light use efficiency model. And I have to give a shout out to Fred Humrick, who's been arguing with NASA for years to make these bands available, the ocean bands available. And they finally are available through Alexei Lyapustin's MAYAC product, but it's taken many, many years, and it's not as easy to access as the standard products. Next slide. So if you look at the bands 1 and 11, upper left, you can see conifers in winter, this is in the boreal forest, undergo changes in their reflectance patterns between summer and winter. And that's due to the changes in the carotenoid pigments. So if you look at the chlorophyll to carotenoid pigments or the chlorophyll carotenoid index, in the bottom left, you see that they track the photosynthetic rate actually quite well. You can do this at the leaf level using needle reflectance in the picture upper right, or you can do it at the canopy level. Next, you see there's instruments on top of that. And there's Chris Wong on the right, who is a Canadian, true Canadian, because he'll go out in a t-shirt in the middle of winter, and if it's above minus 10 degrees, and he was very involved in a lot of this work. Next, please. And when you look at the chlorophyll carotenoid index, this carotenoid index, it shows on the left a very good correlation with GPP for a variety of different conifers, whereas NDVI on the right shows varying and often very poor correlation. It's out of sync, and it sometimes completely fails to respond to the seasonal changes in the photosynthesis or GPP. And this variation is partly due to the fact that some stands have a lot of deciduous component in the background as well, which can drive the NDVI to different degrees. So we do have ways with MODIS of accessing the light use efficiency. Next slide, please. So what I'm going to argue is that with the chlorophyll carotenoid index, sometimes called MODIS PRI, or the photochemical reflectance index, the xanthophyll cycle index, we have a couple of ways of accessing light use efficiency. Now, MODIS can do the CCI. It can't really do the original PRI index. So somewhere in here, I think we've got some opportunities that can help us look at how plants respond to temperature and moisture. And instead of having to parameterize this from meteorological data, we can get the data all consistently from one data source. Next slide, please. So what's really going on here is plants are sessile. They can't get up and run away. So they have to undergo exposure to a lot of different conditions. And some plants are annuals and they go hide. Some plants are deciduous, they'll drop their leaves. Some plants are evergreen and they have to sit there and tough it out under drought, under hot conditions, under cold conditions. So we see varying relationships across these different plant types in terms of how they handle these conditions. But if you think about what plants are trying to do, they're trying to balance the radiation absorption for photosynthesis, some of which also gets turned into heat, with protection, photoprotection under harsh conditions. And they do this, and they release a lot of energy through reflectance and fluorescence. And we can use the reflectance and fluorescence spectra, if you will, to inform us on the basic processes of photosynthesis, the photosynthetic rate, and the degree of downregulation. And the reason this works is in part because if you look at a light response of photosynthesis shown in the bottom right, a healthy leaf under good conditions will have a high photosynthetic rate and a high light use efficiency, which is the slope of the light response curve. And a stressed leaf under extreme temperatures or drought or nutrient limitations or disease might show the red curve, which is a collapse of the photosynthetic rate and a decline in the efficiency with which energy is used. for photosynthesis. So how does this actually happen? So I'm going to take a deep dive for a moment into what happens in the leaf. Next slide. And this is a picture of what happens to the absorbed radiation in a leaf when it's trying to deal with different stressful conditions. Favorable conditions. Under favorable conditions, the light energy is absorbed by chlorophyll, or if it's absorbed by carotenoids, it's passed on to the chlorophyll to drive electron transport and carboxylation. That's the box on the right. That's healthy, normal photosynthesis. Now you can model this with VC max or of Rubisco, or you can use sort of just traditional photosynthesis models to Farquhar-von Kammerer models to look at this process. But what those models historically have failed to do is to show what's going on with the light regulation, which is shown on the left side. Under stress, when the photosynthetic response, light response curve collapses and there's downregulation, what you have next, please. Well, first of all, I should say NDVI is a pretty good measure of the greenness in the absorbed radiation, but it doesn't really tell you how much of that radiation is not being transported to carboxylation under stress. So under stress, what happens is that there's some energy spillover through fluorescence. The fluorescence levels change. The energy gets dissipated to the xanthophyll cycle pigments. In other words, the xanthophyll cycle converts from violazanthin to zeaxanthin, and the energy flows away from the chlorophyll and is dissipated as heat. Now, historically, this was called non-radiative dissipation. It's been called non-photochemical quenching because we're quenching the fluorescence. But it's also, I would call it just downregulation of the photosynthetic system. And so the photochemical reflectance index captures the dynamics of the xanthophyll cycle on the left, this conversion of pigments on the left. So it can track the downregulation associated with these conditions. Now, the chlorophyll carotenoid index captures the carotenoid pigment pool size, how many carotenoids, including xanthophyll cycle pigments, you have relative to the amount of chlorophyll. So sort of the yellow to green ratio, if you will. And so between NDVI, CCI, PRI, and then fluorescence, we have all these ways to access this information. So what happens when you put it all together? That's what I'll be talking about in a moment. So next slide, please. How do we know that this works? Well, can you back up on that? Thanks. Many years ago, over 30 years ago, at Carnegie Institution with a group of people, I did a shade removal experiment. We asked the question, can we see the xanthophyll cycle with remote sensing? So we pictured, we positioned a spectrometer above a sunflower canopy and shaded that canopy since the previous day and removed the cloth, like, pulling out, the old waiter trick where you pull out the tablecloth. If you do it fast enough, all the plates supposedly stay on the table. So you pull out the cloth as fast as you can and you go from darkness to light. And what you see in the reflectance spectra is shown in the upper right, a subtle change in the spectrum in several places. What you're seeing when you look at the different spectrum, the difference between the light and the dark, the light minus the dark state, is a dip around 531 nanometers due to the xanthophyll cycle, and a dip due to chlorophyll fluorescence quenching. That's a double dip. And they're all happening at approximately the same time, which tells you that they're functionally related. And what you can also do, if you're able to do the HPLC on the pigments, the leaf pigments at the same time, which is tricky because you have to work really fast. What you see is that the change in PRI is exactly correlated with the change in the xanthophyll cycle pigments, the conversion from violazanthin to the active forms in downregulation, the zeaxanthin and antheraxanthin, or the deepoxidation state, if you will, or the epoxidation state of the xanthophyll cycles, pretty much is captured by the change in the PRI on a diurnal timescale, or on a short timescale. So this is fundamental information that we can use to assess light use efficiency. Next slide, please. So you can do this on other species. It's tricky, like in this case, we're doing it on pine needles. This is actually a ponderosa pine. I didn't have a good picture, so I stuck it in. But the figures from this are jack pine. This was work I did with Joe Barry and others as part of the Boreas field campaign. And we looked at downregulation in boreal trees, and we saw for sun leaves and shade leaves in the upper left, you can see different constitutive patterns. In other words, different pigment levels in sun and shade leaves lead to different reflectance. And when you do the dynamic dark to light transition, the sun leaf has a bigger xanthophyll cycle pool, a bigger carotenoid pool, and it has a bigger response. And they have similar chlorophyll fluorescence quenching. So they both respond, but they don't respond exactly the same way. And if you look at the bottom right, you see that the shade leaf, as you go from dark to light, the exposure time is on the x-axis and the change in PRI is on the y-axis. What you see is a decline in the shade leaf on the top and a decline in the sun leaf on the bottom over a few minutes. And that changes the delta PRI that corresponds to the xanthophyll cycle pigment conversion. But then the difference between the shade and the sun leaf are differences in pigment pool sizes. The sun leaf having a bigger investment in carotenoid pigments, so it has a lower PRI to start with. So there's a constitutive or slowly changing PRI effect due to the pigment pool sizes, and there's a facultative or a rapidly changing, diurnally changing PRI response shown in the delta PR, which is due to this rapid conversion of the xanthophyll cycle pigments. And both of these are important in photoprotection. They operate on different timescales. Next slide. Okay, so putting this all back together into whole plants, what does this mean for remote sensing of canopies, for example, or leaves? And if you look at, this was work done in a common garden experiment. I think it was 20 or 30 different species, annual, deciduous, and evergreen species on the left. And you see, for example, a sunflower would be an example of an annual, and a coast live oak would be an example of an evergreen species, Mediterranean climate evergreen. And they function very differently in terms of their photosynthesis. In the top panel, you see the annuals have a very high rate of photosynthesis, and the evergreens on the right have a very low rate of photosynthesis. Similarly, in the second panel, the annuals have a high light use efficiency, the evergreens have a low light use efficiency. And the next panel is the fluorescence yield. It's the efficiency of fluorescence, if you will. And that is the very high in the annuals and very much lower in the evergreens. Similarly, PRI is higher in the annuals and lower in the evergreens. So what you see is a parallel pattern of gas exchange, light use efficiency, chlorophyll fluorescence, and the photochemical reflectance index across all these different functional types. And so you have these contrasting photosynthetic properties and corresponding optical properties that can be accessible with combinations of fluorescence and PRI. Now, in this case, the fluorescence is contact fluorescence, a PAM fluorometer, but it informs us of the fundamental controls on the non-photochemical quenching, which is part of the fluorescence signal that we see with SIF. So you can see this. This is a common garden experiment where all the plants are treated the same way. They're all well watered, and basically at midday, this is what they look like to remote sensing. So I think the point here is there's a potential to apply this to look at differences in function within a flux tower footprint or across landscapes or around the whole world. That's where I'm trying to go with this message here. So next slide, please. So the other thing I want to propose is that there's this parallel between the light use efficiency model and solar-induced fluorescence. There's this sort of running argument in the literature, is SIF better than reflectance or vice versa? And I don't think that's a meaningful argument. I think they're very similar and they can inform us of similar things if you use them the right way. So on the left is a sort of cartoon version of the light use efficiency model. GPP is a function of absorbed radiation, but the different slopes refer to the different light use efficiencies, often abbreviated as epsilon. Now, solar-induced fluorescence is basically shown on the right, and it's pretty much tied to absorbed radiation, but the fluorescence yield, which can be captured by that delta F over Fm prime, the PAM fluorescence that I just showed you, or what's called the fluorescence yield, non-photochemical quenching, different terms get used, that affects the slope of that response. So you can sort of imagine, if you believe what I'm saying here, that the SIF and the light use efficiency model might be very similar in function, not identical, but very similar. And we can use this information. This is a powerful concept. Next slide. You hit the return, please. So NDVI can give us this, and we can use carotenoids indices, PRI or CCI, to get to the efficiency, but SIF kind of, I'm going to propose, measures the whole banana, the whole, the combination of absorbed light, and it's also affected by non-photochemical quenching or the changes in light use efficiency associated with the Xanthophyll cycle that are also picked up by these carotenoid indices. So this is kind of a sketch of the relationship in terms of the light use efficiency between SIF and conventional reflectance on the right. Okay, next slide. And this is kind of what's being proposed by the FLEX mission. The European Space Agency's Fluorescence Explorer is going to combine reflectance for things like pigments and NDVI with fluorescence for SIF in the same platform. And this is amazing. I can't wait for this to get up there. I'm going to have to wait a little longer. I think this is maybe next year or so it's scheduled to go up. And satellites tend to never be on schedule. So, but since we don't have this satellite yet, what can we do with multi-scale remote sensing to help us get ready for this future that's coming soon? Next slide. So I'm going to talk now a little bit about SIF. And, this is an old figure from a 2014 paper by Louis Guanter, and using GOM2 data. And it's one of these global assessments of photosynthesis through SIF, relative photosynthesis. And what you see is really interesting patterns, kind of comparable to the general patterns that you might get with MOD 17, but the details are different. And you'll see in the Midwestern US, it's sort of a hot spot of SIF, and it has very high photosynthetic rates. This is the Corn Belt, so it's corn and soybeans, good soil, good climate, tons of fertilizer, lots of water, and it has probably very high photosynthetic rates, so it has very high SIF. And the star is approximately Lincoln, Nebraska, so that's where I'm going to take you now to show you this work that we're doing to look at SIF and the light use efficiency model. Next slide, please. So using aircraft data and ground-based data, we can look at the solar-induced fluorescence that's shown on the left. This is from a paper by Ran Wang, and it shows the radiance spectrum and the SIF spectrum pulled out of that using an ultraspectral sensor. That's the IBIS in the bottom left. And then we also have a Kestrel imaging spectrometer that provides reflectance imagery like the data cube on the right. And that's a forest site in Nebraska. So next slide. So we fly over crops. This is over CSP or MEAD. This is the carbon sequestration program. These are AmeriFlux core sites operated by Andy Suker. And these images are produced by Ran Wang, who's been a good colleague on a lot of this work. And what you see here is just flights over several flux tower sites. I'm going to focus on the CSP3 data, which is an alternate maize and soybean, or corn and soybean, every other year, using standard agricultural protocols for the Midwest. Next slide. So what you see is the field in the upper right, and you see the flux tower in the background. And on the platform is a flux system by JB Hyperspectral. We've been running it, we're in our 8th year now, And I have to give a shout out to JB Hyperspectral. Tommaso Giulita has been great in terms of supporting this. What's nice about this instrument is, combines reflectance and fluorescence capabilities in one instrument. So while we fly over, we can also look at in real, continual time in the flux tower footprint. And so we can get images of fluorescence on the top, and you can see patterns in the field that are not evident from NDVI. You can see regions where there's higher and lower fluorescence. So it's very sensitive spatially. And if you look at the bottom panels on the left is the SIF response compared to GPP through the season. And bottom middle is the light use efficiency model where we're combining absorbed radiation and PRI to capture similar processes. And you see that the SIF and the light use efficiency give us near identical results. If you look at the figure bottom right, they're linearly related, they're very closely related. A bit more scatter in the SIF data, but they're both very good. And so I would say they're almost functionally equivalent here in this situation. And so one of the nice things here is we have an instrument that is now being used by people around the world at flux towers, and maybe we have the hope of standardizing around a common platform and then comparing this to aircraft and satellite data as well as the fluxes. So a little bit of a side thing, a lot of people criticize the light use efficiency model in PRI, and I want to address some of that criticism in the next slide. Also NDVI. If you could advance one slide, please. Okay, so just hold it here for a second. Oh, okay, that's fine. So NDVI does a pretty poor job of capturing GPP because it's really looking at the green canopy structure, the absorbed radiation, if you will. And so we can use it to calculate the fraction of absorbed radiation. And those two lines in the upper right show you a linear and nonlinear fit. They're about the same, but they do make a difference. So how you do the FPAR and APAR calibration matters. And particularly in this case, early and late in the season, it tends to matter. Now, the bottom left is the PRI. In this case, we have scaled PRI from 0 to 1 to turn it into a nice, convenient scalar, like a coefficient between 0 and 1 instead of weird negative values. And it doesn't seem to match very well over the season. And if you look at the fine scale responses of the fluxes to illumination, the noise, which is due to cloudy and sunny days, the PRI kind of goes in the opposite pattern. And that makes sense, actually, because the light use efficiency tends to go up when the sunlight goes down on a cloudy day. So if we multiply basically absorbed radiation derived from the upper right-hand panel with the PRI in a simple light use efficiency model, you get the bottom right. So the problem, I think some people look at PRI alone or NDVI alone and say, well, reflectance doesn't work. But if you put the terms together in the right way and you calibrate it properly, I think it works quite well. Now, there's a part in the beginning where it doesn't agree very well, and I think that's largely because when we installed the instrument that year, so we had a mistake and some people stepped on the plants. It took them a while to recover, and once they got past that recovery, there was an excellent match with the GPP, following day-to-day excursions rather well. So next slide. Some people say, well, what about NIRV? Does that work? Well, yeah, NIRV is in fashion, and it does sort of follow the GPP, but not quite as well as the SIF or the light use efficiency model. And if you look at the bottom right, there's some hysteresis there, and between GPP and NIRV. This is NIRV radiance, and if you use the radiance-based formula, it actually is an excellent measure of APAR. So it's basically measuring absorbed radiation, but it's not sensitive to the short-term changes in light use efficiency detectable with PRI or SIF. It's not as able to do that. So it's useful, and I think it could be a very useful index to use in combination with other information. But that's about as much as I'll say as on that here. But when you integrate NIRV on a global scale over large areas, long time steps, it looks great, kind of like NDVI or fluorescence looks great. Next slide. So, okay, well, this is not all very good. You know, it's easy to do work on uniform corn and soybean fields with simple topography. There's very little diversity. The radiance regimes are very simple because the canopy is very uniform. But the real challenge is most of the world doesn't look like that. You know, there's topography. There's also urban areas where vegetation is mixed with buildings. You can't do flux tower work easily in these highly diverse, topographically diverse, biologically diverse areas with complex radiation regimes and complex fetches and so on. So this is where remote sensing, I think, potentially is really useful. Not only can we look in the flux tower footprint, but we can look at this kind of complexity and see what we can learn, extend beyond the flux tower footprint. So I'm going to show you some results from these two sites, upper right, which is a forest in Nebraska, and bottom right, which is an urban forest. Next slide. So here is the PRI response of individual trees in a forest. And so you can see the landscape, and what you see is the colored blotches are the canopies of individual forest trees, and the red color are low PRI values, or high PRI values, which is low stress, and the blue color is low PRI values, which is high stress. And you'll notice that the high stress or the low PRI tends to be on the top of the hills where it's drier. And the low stress or the high PRI is at the bottom of the hills, the shady parts of the forest. And what we see is a response of individual trees across the landscape to their circumstance. Probably temperature, hotter, it's drier at the top of the hill, and as opposed to cooler conditions. And then there's a lot of individual variation, species to species. This is a collaboration with Sabrina Russo, who has a forest geosite called Indian Cave, Ran Wang as well. And you know, we're seeing detailed patterns of different stress responses in this image. Next slide. And if you look at the actual light response of PRI, and think about those leaf level light responses I showed you earlier. So here we're doing canopy light responses. So some parts of the canopy are shaded, some are sunny. You pick out the pixels at different illumination, and you look at the PRI light response. And it looks like a big blob on the upper right. But if you fit individual tree canopies, you know, lines to individual tree canopies, it looks like a pile of spaghetti on the floor before you boil it. You know, you spill it on the floor, it looks like a mess on the floor, but it looks like a curved relationship, but each individual tree is kind of a linear relationship with different slopes and intercepts. And if you think back to what I was telling you earlier, on the left is sort of an analog to the leaf level PRI light responses that, you know, among different plants, we see differences in intercepts and different slopes that relate to the pigment ratios or the pool sizes, the constitutive effect on the left, and the xanthophyll cycle or the facultative PRI responses on the right. And we actually see that on whole canopy levels when you do light responses. By the way we do this is we use par albedo as a proxy for irradiance, because it's hard to actually know the actual irradiance. But this is how we estimate it in this case. And the interesting thing is, if you look at the slopes and intercepts, how do plants invest in constitutive versus facultative photoprotective processes? There's sort of a trade-off. You'll see as you go up in light levels on the bottom, you see that the pool sizes tend to have one relationship, the xanthophyll cycle response tends to have another. So there's sort of this complementary response. Some plants are doing more with their pigment pools, some plants are doing more with the xanthophyll cycle response, is our interpretation here. So we're starting to see plant level responses. And the only reason we can do this is because Sabrina Russo knows the identity of every single tree in that plot. Being a forest geoplot, they measure that stuff. So next slide. You take the same approach and you go to an urban campus, which you also can't measure with fluxes, but we can look at SIF or PRI. And what you see is similar sort of PRI light responses in the upper right-hand corner. And the SIF responses are also quite interesting. The idea is increasing light should increase SIF, but the slope of these responses should be related to the fluorescence yield. And we see species level differences when you fit individual species to this, what looks like a big mess. You see consistent and significant differences between evergreens and deciduous. And also there's one plant that really stands out, which is the green ash, as being different both in its PRI and SIF light response. That's a tree that is being attacked by the emerald ash borer. And what I think we might be seeing is early ash decline. It may be invisible to our eyes, but you can see it with these light responses of downregulation. Those trees were subsequently removed by the campus in the next year, and so they're gone. But we think we saw the beginnings of that infection possibly on those trees. So we can see early detection, pre-visual detection of individuals that are not functioning well in a landscape with these methods. Next slide. So what I wanted to sort of emphasize here is we don't have all the satellites yet. We have some hyperspectral satellites. The FLEX satellite is coming. I think it could be very useful. We have the SBG that we hope will be coming, a hyperspectral satellite. Meanwhile, we can do a lot of work with aircraft and proximal remote sensing. I've just shown you a bunch of work that we're doing with aircraft, but I think a community like this can do a lot together to try and pull together not only information on fluxes, but if we can be consistent in how we measure and report our optical data, I think we could do a tremendous amount within the flux community to advance these questions. Next slide. So I started with a rainbow. I'm ending with a rainbow. I think, rainbows are beautiful, but they also remind us that the interaction of light, different spectral qualities of light, the different colors, if you will, and matter, the atmosphere or the vegetation can tell us all kinds of stuff that can inform us about fluxes. Next. Optical information adds complementary information to our understanding of carbon flux. At its best, I think it's about as good, as accurate as flux measurements themselves. We're within the error of flux measurements. So what's the truth? I don't know. I mean, I think the idea is if you combine these things, it's much more powerful. And I think the light use efficiency model gives us results that are comparable to solar-induced fluorescence, very good tracking of GPP. And spectral information can add insight into contrasting function. In other words, functional diversity. It's an aspect of biodiversity that we often don't think about, I think, in the flux community. Within the footprint, can we see that diversity in function? And can that inform our understanding of whole ecosystem fluxes? Across biomes, do we see, across flux tower sites, do we see that same functional diversity play out? And I think the answer is yes, if we could do that work that way. And I think this is important because functional diversity, the way plants do photosynthesis, it's related to vegetation productivity, obviously, GPP and NPP. Plants just have different strategies for getting there, and that understanding that is important. But under stress, flash droughts, temperature change, climate change, et cetera, understanding that functional diversity is really important because it's part of the resilience that ecosystems have, to change or to disturbance. So I think this is a missing part of, what's been missing in a lot of flux measurements is digging into that footprint. And I know people are working on this, but I think this is one way we can combine remote sensing with flux measurements to really add this kind of information. So thanks very much. One more press of the key. Thank you for your attention. I appreciate your time today.
Thank you so much, Dr. John Gamon. So if you have any questions, please, unmute yourself and just ask John, or you can type your question on the chat. So I have one question. This might be a little big question for this, but so I agree with what you said about, there's no single way best approach for everything in this proximal remote sensing. There are many different types of proximal remote sensing measuring one feature. But that also poses, I think that also poses us kind of issue or challenges in kind of standardizing the measurement across using different approaches. So do you have any insights on how we should approach to make it,
Well, I have a few thoughts. That's a good question, and it's one that comes up over and over again. That was one of the original goals of SpecNet, was to try and come up with standardized protocols. And we found it impossible to do because we had no funding to enforce standards. And in the absence of funding, people around the world are going to use whatever's available. Usually it's what's cheapest, what they can afford, and what's suitable for their particular ecosystem. So a method that works on a grassland or a crop may not work on a tall forest. And so you have apples and oranges, alphabet soup of methods. So standardization has been elusive. I'd say that in a way, the European community has progressed further than North American. The European Space Agency with the FLEX mission has supported the use of JB hyperspectral sensors at a bunch of flux tower sites. I don't know how many there are around the world, but I think efforts like that to adopt a particular instrument, and I'm not saying that's the only instrument to use, but when we can identify something that's available that is being used, I think that can help. And what's largely missing is sort of, in addition to the instrument standardization, is standardization of methods and standardization of like data management approaches. So I would love it if AmeriFlux would take on this problem. I think it's maybe too big a problem. It's for AmeriFlux, it's maybe a bit beyond that, but I know Gilberto has thought a lot about incorporating optical data with flux data. And I think that's a good direction. I think AmeriFlux could help. I think that solving the data standardization, and I don't know who would want to pay for the data systems. Meanwhile, JB Hyperspectral collects data from its instruments and can help a lot with provision of standardized software, open source software. Their stuff is open source. They're very helpful. So I think it's a combination of efforts like that that are currently moving us forward. And within SpecNet, we're talking about this issue a lot still. And so if you're interested in this issue, I would encourage people to reach out to SpecNet as well. There are separate efforts at standardization around the world, but the problem is they're separate efforts. So ICOS has one effort, Eurospec had an effort, different groups had efforts. And because they're funded by different places and they come and go, and technology changes, it's very hard to solve this problem. But I think there are bits and pieces that can help, and maybe we can focus on those as a starting point.
Thank you. So Dennis, I see you raise your hands.
Yeah, wonderful talk, John. I really, really appreciate it. I'm glad you actually mentioned NIRV. But I guess I have two questions why I kind of like it. One is a stronger signal in SIF, and the other one is we work a lot with tules and grasslands that have a mix of green and dead vegetation. So there's these plant elements that are absorbing light, but they're not using it for photosynthesis. And we actually find that NIRV does a really good job of helping us simulate GPP pretty accurately. So I was just curious about your comments, essentially. And would you revisit NIRV as we go to other types of landscapes besides crops and evergreen forests?
I haven't used it in a lot of different sites, so your insights are, I can't really comment too much on that, except that I know there's a lot of cases where it works really, really well. And to me, it's partly the temporal and spatial scale that you're operating at might be part of that. But I think it's, let me ask you, are you using the radiance-based or reflectance-based NIRV?
The radius space, because we have it on our tower, so we can do that. So
Yeah.
But you gave me a good hint too. I mean, a while back, I gave a talk to Young Reels Lab way back when, and I was trying to play with PRI and NDVI at my tower and compare it with the fluxes. I was getting noise all over the place. And then he had just come back from sabbatical with Joe Barry and kind of pointed me to NIRV. But what I haven't done is try your product of PRI and NDVI. So I'm going to go back and relook at that. I think that was really see that from you. Appreciate
Well,
it.
you can, with any of these approaches, you can get really good results or really lousy results depending on the details. And so in terms of the light use efficiency model, not only how you parameterize it, but how you calibrate it. And in our case, we calibrate it against direct measurements. Some people use radiative transfer models. Some people use other methods that are indirect for estimating FPAR, in a calibration. They give different results. And, I'm not particularly fond of the way Modis does it, for example. I think one of the big problems with the Mod 17 is the FPAR, APAR, LAI products that go into it are flawed. They certainly don't agree with independent field measurements that I've done. And they're based on, radiative transfer and theoretical approaches, which work pretty well maybe at a global scale, but I think they fail to account for the structural differences and physiological differences in different vegetation. So, I mean, your question about NARV is a good one. I think it's, to me, it looks like a really good measurement of APAR. And if you're not limited by temperature or water or nutrients, it's pretty much the whole thing. I mean, you don't need a whole lot more. But I think when you have a lot of stresses and like in conifers, for example, in cold temperatures, extreme drought, it may not work as well. And because it doesn't seem to capture that efficiency term. So, under different circumstances, it might behave slightly differently in terms of GPP. And if it works great for you, I think that's great because it's a good signal and it's, kind of like NDVI combined with a light term, in the radiance. So that's kind of like the light use efficiency model. It's interesting that it behaves rather similarly in our analysis and provides a really good estimate of absorbed radiation, which is in some cases most of what you need to know.
I mean, if I run it through my CanVeg model for irrigated crops, it works really well from first principles, essentially. Yeah. But I agree with you. I'm still trying to figure out the best way to deal with Tonzi Ranch with water stress. And so that's why I'm still curious about PRI and these other indices. So thanks. All the others have questions.
Thank you. And John Ho, do you have questions for John?
Yeah. Can you hear me?
Yes.
Yeah. Hi, John. I'm from Korea. Yeah, here, as you see in here is the night. Thank you. Thank you for your presentations. I'm super enjoy. Yeah, my question is about the water stress or water effect on the sieve. As you mentioned, the sieve is the trace of the right response. And however, there's some study are being published that suggest the sieve may be deeply related with the water stress or water storage. So the first question is, do you agree with this? And if so, what kinds of physiological effect or mechanism do you think water has on the water as on the water affected by a sieve affected by water?
Yeah. Okay.
As the physiological or mechanism. So, yeah, thank you.
Well, that's a good question. And I have to say that I haven't done a lot of SIF measurements under drought conditions. My focus so far and what I presented was mostly on well-irrigated crops or crops in a rain-fed environment where there was not a serious drought. But to the extent that, I have looked at it. There is a slight sensitivity to the yield, the SIF yield. In other words, non-photochemical quenching or the downregulation that I've been talking about, it is sensitive to that. Now, in my system that I just presented, there's not a lot of that stress. It's a fairly small component. If you go to a system where that stress is big, I would expect it to play a bigger role in the CIF signal. The CIF yield can be variable in different conditions. It's affected by stress. Now, exactly how that plays out across different ecosystems having contrasting structure and physiology, and under different stresses, water stress, nutrient stress, temperature stress, I don't think that work has been done. Individual studies have been done looking at elements, but I think that's what we don't really know. We have a lot of site-based work that shows sometimes evidence for stress effects on CIF. And then you have global or continental CIF studies from satellite that suggests CIF is very strongly related to absorbed radiation. And the problem is at that scale, we're not going to see all the fine scale regulation. It's the sort of integrated argument that I made with NDVI. You can make that for CIF. It's at that scale, you're not going to see the fine scale regulation and the effects of the down regulation on the CIF signal. So I believe it's there. I believe it has to be there just from basic principles. Demonstrating that is tricky, and demonstrating it in a uniform way across vegetation types, ecosystems, and biomes I think, still needs to be done. So, I mean, some of this can be done through experimental work or through networks like AmeriFlux, and maybe SpecNet, other networks. But I think part of this maybe can be addressed through modeling as well. I think there are some really good efforts to integrate photosynthesis modeling with radiative transfer modeling, the scope model. There are other ways that might also inform this. So I have to say, I don't really have a good answer for you, but I think there has to be an effect of water stress on CIF, and it would be through the yield and also through the absorbed radiation, because plants, depending on what kind of plant, are going to change their structure. So exactly how that happens might be different for different vegetation types. I hope that answers your question.
Yeah, thank you for Gamon. Yeah, it is too tricky. Yeah, I will focus on this topic during my PhD course. And yeah.
Well, I'll be interested to hear your answer.
Oh, for me, I think it will be affected by the MPQ. I mean in my just my think or hypothesis or something like that. So when I think the plants want to maintain their high efficiency for the energy partitioning part, as you know. So if the plant got a higher energy or some kinds of got some stress case, I guess the plant will be partition that energy to the MPQ first, and then PQ, and then the fluorescence is the last step. But sometimes, so uh, give me a second, please. I'm nervous. Honestly,
Yeah, well, it's interesting. Just a brief comment is you're thinking it in terms of the fundamental mechanisms, which is really good. And tying that back to the observable reality in the optical signals is kind of tricky. I mean, it takes a lot of careful work. And so it's a challenge what you're thinking about. I think that's a great topic, though.
Yeah, thank you, Gamon. And my second question is about the next Flux Optics meeting. I super expect the next meeting. So, is there any plan or some kinds of that for next meeting? I cannot join last meeting.
No.
I will, yeah.
You mean, are you talking about within AmeriFlux or SpecNet or a joint meeting or?
The I think second one, the in last year's case, the
Okay.
you guys met in the with the Troy maybe.
Okay.
I mean, so I'm just curious, is the kinds of meeting will be held
Yeah.
or not in this
Well,
week in this world soon?
that's a good question. I mean, I imagine there'll be another SpecNet meeting probably sometime this year. We have one or two a year typically. It's a volunteer effort. So we're a nonprofit, so it's when the spirit moves us. But I think having people interested will drive us to have more meetings. And there's also opportunities for helping to organize meetings, or they're kind of fun to do. And with Zoom, we can do this and have people all over the world, and it's much easier. And it doesn't cost us any money compared to trying to meet at AGU, which is what we used to do. So yeah, I think we want to keep having meetings. And so I'm glad for your interest, and please reach out to, SpecNet or AmeriFlux folks as you feel you can to express your interest in these ideas.
Yeah, thank you. If it is held, I will look at the fund for my PI Younger. So I will expect you meet you in person soon.
Okay, great. Thanks for
Thank you.
thanks.
Great. So I do see one question in the chat, if you have time to answer, John. So how can we integrate data from different vegetation indices, like your example of NDVI and PRI, to get GPP as light use efficiency model?
Yeah, that's a good question. And one way, the way I've done it is statistical. You do a whole bunch of things and you look at all kinds of combinations. You can do it purely statistically, or you can do it based on a mechanistic understanding of what the indices do for you. And I think with the kinds of statistics we have, and machine learning and other kinds of tools, it's possible to automate a lot of this. But many years ago, that was something that I would have done manually, and it's just very time consuming to do that. Now, some people are very critical of the index approach and say, well, you should use full spectral information and maybe something like PLSR, if you have it, to inform you of the wavelengths of interest, and that can drive how to do this, but you have to have full spectral information. So we don't always have that for our sites. What I provided was sort of a framework using MODIS data and comparable, similar results from aircraft and field data, just to show that these methods can work in the context of the light use efficiency model. So I think that that would be one kind of conceptual framework that I would find useful in organizing your thoughts as to which indices might work. And a good question is, what role does NIRV play, or what role does MODIS PRI play if you don't have PRI? And these differences in these questions are kind of interesting, but they're also complicated because they're not exactly the same as something else. NIRV is kind of like NDVI times irradiance. It's kind of like an APAR measurement, but it's not exactly that. Maybe it's very similar, functions that way. CCI is kind of like PRI. The MODIS PRI is kind of like the traditional PRI, but it's actually different wave bands, and it's very sensitive to pool sizes, and it may be not equally sensitive to the Xantheville cycle under those conditions. So, these are complex questions. I think maybe this is something we could follow up offline, because it's kind of a big question, how to do these kinds of studies. It's a good question, though. And I would want to, I hope that answers your question. I want to point out too that Fred has a comment that I think is important, that we've lacked hyperspectral satellite data at a global scale, but we now have the PACE satellite, which was designed for ocean color. But Fred has done a lot of work to make the PACE data available on land, sort of like he did with MODIS data, and he's been arguing for that. and the terrestrial surface information should be available soon. So we do have some new sensors that are just becoming available, some of which were in one of my slides. But I think it would be good to look at PACE, because it is one of the first true satellites, or satellites that can provide us with the original PRI formula. So we can look at different formulations with hyperspectral satellites and help address the index problem. This helps the standardization question, because up until now, we really haven't had a standard satellite product even, but we're starting to have those. And maybe that can help drive the instrument, the proximal, the aircraft standardization as well. At least the satellites will be done consistently, which we do not have with proximal sensing or aircraft sensing. So I think that's an important point. And thank you, Fred.
Awesome. Are there any other questions for John? Okay, I think, yeah, it's past 10 already, so maybe we can wrap up, or if you want to stay, hang around more and talk to John, please do. But thank you, Dr. John Gamon, for sharing your precious insight with us. Today's presentation is recorded and will be uploaded to the AmeriFlux YouTube channel in the coming days. So with that, thank you for joining us today, and see you next time. Thank you.
Thank you as well. And I appreciate, the audience. You've been a great audience, and thanks for your time.
Okay. Thank you, John. That's really a great talk, and I really liked your talk, and I do have a lot of questions regarding, because I have done site-specific measurement of some of the proximal remote sensing techniques.
Sure.
So, but I don't know if you have time to talk more about this right now.
Sure.
Or
If you want, I can stay on for a bit and answer, talk with you or answer additional questions. I'd be happy to do that.